I just point out, I just learned a few minutes ago that the Lasker Awards this year were given to Max Cooper and Jacques, Cooper for discovery of T cells, back a long time ago. So it's about time the importance of T cells gets recognized. So probably don't need this. So what I'd like to do today was asked to come and give you a brief review of where the field of checkpoint therapy in treating cancer stands. And obviously there's going to be a little bit from my perspective, and then show you the directions in which I think it's going to go in the future. And then mostly towards the end, I think what we're learning recently is, there's two major checkpoints in development now, are approved for many indications, still in development for others, CTLA-4 and PD-1, and they're remarkably different. And so they're treated very often as if they're the same, but I want to show you today how remarkably different they are in their mechanisms of action, and why this realization, I think, is going to open up a lot more areas of investigation in this area. So the work today, I've had a lot of people working with me over the years. I didn't really show it. But the work I'm going to show today is largely the product of Spencer Wei, a very talented postdoc in the lab, with collaborations from Pam Sharma. As always, we work together, and some computational stuff from Donna Pear, and from the Immunotherapy Platform at MD Anderson. So I'd like to start with this, where are we now? What does checkpoint blockade do? This is Sharon, a woman in Santa Monica, California, who was diagnosed in about 2000 or so with stage 4 melanoma, and she'd failed everything, including high-dose IL-2, and dendritic cell vaccine, a bunch of chemotherapies and stuff, had failed them. And Tony Rebus was her doctor at the time, and he said, well, we've run out of things in the kit. We got this new thing called ipilimumab that hasn't been in people before. We don't know the risks, but, would you like to take it? She said, I'll do anything. I just want 3 months to see my son graduate from high school. Well, that was in 2001, and she was on, as I said, she was on the phase 1. She got a single injection of 0.3 mg per kg, 3 mg per kg of ipilimumab. And you can see her lung mass there in the pleural effusion. About 3 months later, it was completely gone. I visited Tony in 2011, which is her CAT scan on the right. I was visiting him 10 years later, and she's still fine without any additional treatment. And she's still fine now. Every time I talk to Tony, we keep up with her. So she's almost 19 years out now after a single injection of anti-CTLA-4. So that just shows the durability that you can have from these responses. And I'll mention it now, and we can talk about it later if you want, but I do know that patients like her and several others that I've gotten to know pretty well do not like the idea of you telling them they've got a manageable disease. I think after 5-10 years, patients deserve to be able to say, I'm cured, so they don't have to look over their shoulder for the rest of their lives wondering when this is going to come back, because I'll show you in a minute, you really don't have to worry about this. Well, how does this work? Well, I'll show you. It seems complicated, but it's not that complicated. But the bottom line is, when we started on this work, I was really interested in how T cells, worked. I mean, when I took a course in undergraduate school just after the original work was done by Max and Jacques, all we knew is T cells are these cells that go all over the body and look for stuff and deal with it. They go through your tissues. And I asked the professor, I said, well, how does that work? And he said, well, we don't have any idea. And he said, but I'll tell you, I don't think there is such a thing as T cells. I think they're just other cells that have picked up an antibody molecule, and they've gone someplace because nobody could figure it out. Anyway, a lot of things that people, obviously we had tried to mobilize the immune system to treat cancer for many years and had not really succeeded. And I began to think that that's because people were getting over the tips of their skis, if you will. They were trying to do stuff that they didn't understand. Like they could, IL-2 is T cell growth factor, so let's just pump people full of it, and that ought to get their T cells to, kill cancer. It makes them real sick, and sometimes actually cures their melanoma, but nobody's ever done a trial that provides any evidence, for example, that has anything to do with T cells whatsoever. But anyway, ultimately, a lot of things didn't work. And so I just decided we really need to understand in detail how T cells work. Because we actually worked out the protein structure of the T cell receptor in 1982, and it became clear by the late 80s that was not sufficient to activate T cells. There were other signals called costimulatory signals. And so one of the things that we realized over the years is that tumor cells are largely invisible to the immune system because they can't give that second signal. They don't have the ability to provide the B7 stimulation of CD28 on the T cell to get it going. And so the immune system doesn't know they're there. We proved that in the early 90s by putting the B7 gene into tumor cells. They immediately get rejected. So they had plenty of antigens. It's just the immune system couldn't see them. And so I'll try to put this together now to make some sense. But anyway, so the tumor cells, for the reasons I mentioned, I'll show you a little bit more graphically, cannot provide both signals. So the only way the immune system knows they're there is for there to be some death of tumor cells, some inflammation that brings in dendritic cells and other cells that can reprocess the antigen from the tumor cells on their surface in the context of B7 so that you get the CD28 signal contemporaneously, and that tells the T cell to go. It actually turns on a program that says divide like crazy, generate the differentiate, generate the army that we need to go out there and deal with some sort of threat. And so, but we learned in 1992 or so that there was a homolog to CD28 called CTLA-4. A lot of people thought it was another CD28, but my lab and Jeff Bluestone showed that, in fact, it wasn't. It was a break. But it's induced. There is no CTLA-4, first of all, in a resting T cell. T cell constitutively always has T cell receptor and CD28. So if it gets activated by signals through TCR and CD28, that turns on this GO pathway that I just told you about of cell cycle genes and cytokine production and proliferation, but it also turns on the CTLA-4 gene. It's hardwired into the system. It has nothing to do with tumors. It's just hardwired into the immune system so that something can stop that proliferation, because you can't have T cells dividing at the rate of bacteria, which they are at the peak of their expansion phase. They're dividing about every 6 hours. You can't have that going on for very long. And so the purpose of CTLA-4 is to be induced and come up, and after a time shut off costimulation and keep new cells from being recruited, and keeping the cells that run down after a while and need rejuvenation through costimulation to come back up. And so that can then frustrate the ability of the T cells to go out and kill tumor cells, especially because in this model, the tumor cells gotten a head start by being invisible so long. Bacteria, things like that, don't get a head start because with infect cells, that induces the innate immune system to immediately know they're there. So anyway, we had the idea, well, let's just take the brakes off and keep the T cells going for as long as we can. We could keep them going as long as we need to get rid of tumor cells. And so I'm not going to show you the experiment here, but well, I am, actually. I thought this was going. Anyway, this is the first experiment we did in mice. I'm sure everybody's seen these, but the black line is a tumor. It really doesn't matter what it is. It's a transplantable tumor growing on the flanks of mice. We inject antibodies that block CD28 co-stimulation. The tumor grows faster, actually, a little bit faster, telling you that there is kind of a nascent immune response, but it can't get the job done. But if you inject anti-CTLA-4, the tumor grows for a little bit and then gets rejected. And the mice are permanently immune to re-challenge them. You could, 2 years later, challenge them with a tumor again, and they'll reject it with no therapy. And so, we got this result. I was really astounded, and we were really onto something because of all the molecules that are involved in initiation of immune response. Blocking this one molecule, CTLA-4, turns that black line into the red one, and the tumors just melt away. This didn't always work. It worked in tumors that we finally figured out after a couple of years of doing every tumor model we could think of or acquire. It works very well on tumors that have that are chemically induced, methylcobalamin-induced fibrosarcomas, other really, really invasive, lethal tumors that we know now have high tumor mutational burdens. Does not work so well by itself against tumors with lower burdens, although I don't have time to show you today, but we've been able, by combining this with radiation or chemotherapy in some cases, or with other checkpoints.